Hi dear students. Today's session, let's begin with Archaebacteria. In 1996, scientists decided to split Monera into two groups of bacteria, Archaebacteria and Eubacteria. Because these two groups of bacteria were different in many ways, scientists created a new level of classification called a domain. Now we have three domains, bacteria, archaea and eukaryote. Archaebacteria are the oldest organisms living on Earth. They are unicellular prokaryotes without cell nucleus and any other membrane-bound organelles in their cells and belong to the kingdom Archaea. They were first discovered in 1977 by Carl Rose and George E. Forbes and classified as bacteria. Most Archaebacteria appear like bacteria when observed under the microscope. However, they are quite different from bacteria and eukaryotes. Archaebacteria are found in very harsh conditions such as in the volcanic vents or at the bottom of the sea. They can easily survive in such extreme environment at sea vents releasing sulfide-rich gases, hot spring or boiling mud around volcanoes. So the kingdom Archaebacteria is a large group of primitive bacteria having unusual cell walls, membrane lipids, ribosomes, and RNA sequences, and having the ability to produce methane and to live in anaerobic, extremely hot, salty, or acidic conditions. Some of the first Archaebacteria were discovered in Yellowstone National Park's hot spring. They are structurally simple but biochemically complex. They will live in extreme environments and all are unicellular and there is no peptidoglycan in their cell wall. Some have a flagella that aids in their locomotion. Most of them don't need oxygen to survive. They can produce ATP from sunlight and they can survive enormous temperature extremes. They can survive under rocks and in ocean floor vents deep below the ocean surface, and they can tolerate huge pressure differences. Let's see the structural properties of archaea. They are slightly less than 1 micron long. A micron is 1 by 1000 of a millimeter. In order to see their cellular features, scientists use powerful electron microscopes to view it. The shapes can be spherical or bowl-shaped and are called coccus, and others are rod-shaped, long and thin, and they are labeled as bacillus. Some archaea have flagella, which are the hair-like structures that assist in movement. They can be one or many attached to the cell's outer membrane. Protein networks can also be found on the cell membrane, which allows cells to attach themselves in groups. Cell features. Within the cell membrane, the archaea cell contains cytoplasm and DNA, which are in single-looped forms called plasmids. Most archaea cells have a semi-rigid cell wall that helps it to maintain its shape and chemical balance. This protects the cytoplasm, which is a semi-liquid gel that fills the cell and enables the various parts to function. The molecules that make up cell membranes are called phospholipids, and this acts as a building block for the cell. In archaea, these molecules are made of glycerol ether lipids. The ether bonding makes it possible for archaea to survive in environments that are extremely acidic or alkaline or that have great extremes in temperature. Let's see the classification of Archaebacteria. There are mainly 5 phylums in it. The first one is Archaeota, then Archaeota, 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 Archaeota. The first one is the Archaeota. It means scalloped archaea because they are often irregular in shape. All Archaeota synthesize a distinctive tetraether lipid called crenarchaeol, originally containing thermophilic and hyperthermophilic sulfur metabolizing archaea. Recently discovered crenarchaeota are inhibited by sulfur and grow at lower temperatures. These organisms stay gram-negative and are morphologically diverse, having rod, cocci, filamentous, and oddly shaped cells. Let's see the examples of Archaeota. The best characterized member is the Sulfolobus solfataricus, isolated from geothermally heated sulfuric springs in Italy, and grows at 80 degrees Celsius, and it can survive a pH between 2 and 4. Other examples are Pyrolobus fumari, Sulfolobus solfataricus, and Sulfolobus acidocaldarius. The next phylum is Archaeota. It has diverse 7 classes in it: Methanococcus, Methanobacteria, Halobacteria, Thermoplasmata, Thermococci, Archaeglobi, and Methanopyri. And it consists of 9 orders and 15 families. On the basis of habit, they are divided into the following. The first one is methanogens, the second one is extreme halophiles, and the third one is sulfate reducers and many extreme thermophiles with sulfur-dependent metabolism. First, let's take into the halophiles. Halo means salt and phil means loving. So, halophiles means salt loving. The halophilic organism requires salty environment for their survival. They are found in salt, lakes and areas where evaporation of seawater occurs, such as the Great Salt Lake in the US and in the Dead Sea. It can live in water with salt concentration exceeding 15%. The ocean's salt concentration is roughly 4%. Examples of halophiles include Halobacterium, which includes several species that is found in salt lakes and high saline ocean environments, and it includes Halobacterium salinarum, Halobacterium denitrificans, and Halobium. The next one is the methanogens. Methanogens are microorganisms that produce methane as a metabolic byproduct in anoxic conditions. They are strictly anaerobic organisms and are killed when exposed to oxygen. They reduce carbon dioxide using hydrogen and release methane in swamps and marshes, that is called marsh gas. They live in mud at the bottom of lakes and swamps because it lacks oxygen. They are also found in the gut of some herbivores like cows and humans, dead and decaying matter. They are added to biogas reactor for production of methane gas for cooking and sewage treatment plants. Examples of methanogens include Methanopholis aquamoris, Methanopholis ethanolicus, and Methanopholis formosanus, Methanopholis liminatans. The next one is a thermophilic and sulfur-reducing algae. All thermophiles require hot water but differ in other habitat needs. Some thrive in only acidic water and others require sulfur or calcium carbonate, and some others live in alkaline springs. Depending on characteristics which it may possess, they can be described with more specific terms such as thermoacidophile, that is heat and acid lover. They have both aerobic and anaerobic species. The next one is hyperthermophile, that means it can survive extreme heat, so they are termed as extreme heat lover. An example of that is the Pyrolobus fumari, currently holds the record for high temperature growth. It can grow in temperatures up to 113 degrees Celsius. Here you can see the image of halophilic bacterium, thermophilic ones, and the methanogenic Archaebacteria. The next phylum is Archaeota. The name is derived from the Greek noun kor, or kor means young man, and the Greek adjective archaeos, which means ancient. They are also known as Synarchaeota. These Archaeota have been found in high temperature hydrothermal environments, that is in Yellowstone National Park. They were seen in springs with a pH range of 5.7 to 7.0. The next one is the Thomarchaeota. Here, Thomar means wonder. It was proposed in 2008 after the genome of Craniorcheta symbiosum was sequenced and found to differ significantly from other members of the phylum Craniarchaea. All organisms in this is found to be chemolithoautotrophic ammonia oxidizers and may play important roles in biogeochemical cycles such as nitrogen cycle and the carbon cycle. Next phylum is the Nanoarchaea. The Greek meaning of this is the old dwarf. They inhabit high temperature environments with an optimal growth of 90 degrees Celsius and are highly unusual because they grow and divide on the surface of another archaea, Ichnecoccus. Nanoarchaea, which were discovered in 2002, contain both the smallest non-living cell and the smallest known genome. Members of this phyla have not been detected in pure culture. Let's see the importance of Archaebacteria. The archaea is able to produce extremophilic enzymes and they are resistant to heat or to extremes of acidity or alkalinity. For example, thermostable DNA polymerases from Pyrococcus furiosus is used in PCR, and enzymes produced by Pyrococcus such as amylases galactosidases is used in food processing at high temperature. Methanogens archaea also play an important role in sewage treatment. They carry out anaerobic digestion and produce biogas. They are also required for the extraction of metals like gold, copper, cobalt from their ores. They play important role in carbon cycle, nitrogen cycle and sulfur cycle. They also help the researchers because they have the ability to tolerate extreme conditions that help the researchers learn about the climatic conditions, environment and their survival on ancient Earth. Archaea host a new class of potentially useful antibiotics